Hello friends, good evening. Today we are back with a new video. Actually, this is a start. This video is the starting point of a new series. Actually, I wanted to make one series, so I thought that regarding the videos of the funguses, I should come to a video, make a video which is the background of this virus, this fungal videos, the producing of fungus videos. How I test the videos in the fungus or how I see the fungus. So for that you need an arrangement, and that arrangement is called a microbiology lab. So today the topic is around how to make a microbiology lab. That is the our topic, or rather how to make a economic microbiology lab. Because microbiology lab is there is no limit. The sky is the limit. So there is no limit, but our pocket is very limited. So regarding this microbiological lab, making a microbiological lab, we have a very pocket limited budgets. So for that we need some requirement, we need some instruments, we need some chemicals, we need some apparatus. So that we are going to explain in this series of videos. So how economically we can make these videos, so that we are going to make in this series. So the first part of the series is the main thing, that is the cabinet. We are now going to make a video on this biosafety cabinet, which we say is the first requirement of a microbiology lab. If you don't have a biosafety cabinet, you cannot perform any histological work or any mycological work. You cannot extract the fungus from the culture media, or you cannot extract the fungus from the host and put it in the culture media. That you cannot do. So for that we need a very economically microbiological cabinet. So what is it? A cabinet is nothing, just a closed environment, a closed, what should I say, closed, disinfected cabinet, where you can sterilize or disinfect the things, keep things disinfected and sterilized without any contamination. That will be a closed cabinet. So that we are going to talk about in this video. So let us proceed regarding this video. In the next part of the video, we will see what is a cabinet and what is inside the cabinet. What we require, the basic tools of the microbiology. Then that we will discuss. These tools are not very costly, but little investment is necessary. So that we will talk about. So that we will be going to discuss in the next part of the video. So please be with us to see this video and this series of the videos. So friends, this is the cabinet, as you can see. So now we are going to open the cabinet. As you can see, it is an wooden cabinet. Above you can see there is a rack, and this is a cabinet. This is around 2 feet, 3 feet by 2 feet in depth, and it is just a wooden cabinet. It's a simple wooden cabinet with two doors. And when I'm opening the door, the light is switched on. This isn't nothing, just but an automatic setup of switch which is above there. So this is the switch. So if you push the switch, the light will off, and if you lift the switch, the light will be switched on. This is just a common, what would I say? It's a fluorescent tube with a small 1.5 feet fluorescent tube, and behind that you can see a transparent light. You can see that is the UV light. So this is also necessary to keep the cabinet sterilized. So when you finish the work, or you just before the work, you have to switch on this UV light, which I am going to switch it on now. You will be able to see. When I will switch it on, you will be able to see, but that this light should not be visible, because when you close the cabinet, only you can switch on the light. So that will give you a violet UV light, so you can sterilize this cabinet, or there you can disinfect the cabinet. Now we are going to look out the things what are there. So you can see the main thing is there. You can see this is a, first of all, this is a solution of 75% IPA and 25% water. This is a very distilled, this is just a distilled water for washings, for your hands. You can wash the pipettes and all that. So these are the culture or the sterilized pipettes. As you can see, these are sterilized pipettes. These are also available. These are called serological pipettes, disposable serological pipettes. This is available. This is around 1 to 10 ml. From 1 ml to 10 ml, this is a 10 ml serological pipette. So this is also available. And now behind that you can see there is a remove this. You can see this is the pipette rack. This is the main thing for the microbiology lab. This is the pipette auto pipette rack. These are the different kinds of auto pipette. I have actually three types of three kinds of auto pipette. This one is from 0 to 200 microliter auto pipette, or rather from 2 microliter to 2.5 20 microliter auto pipette. And the next one, this is from also from 2 micro ml to 1,000 micro ml auto pipette, variable auto pipette. This one is from 1 ml to 5 ml auto pipette. This one is a fixed 1 ml auto pipette. This one is a fixed 1 ml auto pipette. So this is another 1 ml to 5 ml auto pipette. This is also a fixed auto pipette, 1 ml, 1000 micro ml fixed auto pipette. So these auto pipettes are required for different kinds of solution, taking the solutions of the fungal media or the culture media. These are required. So just aside of this, these are the auto pipette tips. This is the 1 ml to 5 ml auto pipette tip. This is 2 micro ml to 20 micro ml auto pipette tip. And this box also consists of, I can see the closer look, you can see these are the 1 ml micro pipette tips, to 20 micro ml to 1000 micro ml tips. These are the smaller ones. These are the smaller one tips. You can see this is for the 2 to 220 micro ml, 200 micro ml tips. So that behind that is just nothing just but cotton plugs for making the cotton plugs. As you have seen in my previous videos, I used to make cotton plugs. So those are the cotton plugs. This is a dry cabinet. This is actually, if I switch on the light inside, you will be able to see this is the drying cabinet. You can keep the slides here inside to dry the cabinet. So this these two has a exhaust filter, so you can exhaust fan, so you can dry the filter, dry the slides and all those. You can dry the slides, you can dry the for staining and all those. You can ship. These are the two test tube racks. This is a simple test tube rack. This is a microcentrifuge test tube rack, and this contains the microcentrifuge tubes, the small microcentrifuge tubes. If I open the flask, let us remove this and let us open this container, and you can see these are the microcentrifuge tubes. So these are the 2 ml microcentrifuge tubes. So that is also available in the market. So these are some small flasks needed for mycotoxin extraction, and this is a magnifying glass. So this is necessary for macro investigation. This is just nothing but a magnifying glass. You can put on and it will be switched light. You will put a light and you can just draw it like that, and you can see and make a you can watch something from there, and that will be a good low magnification type. And those are the tissue papers inside. This is the tissue paper you can use for plugins and all that. That is an electrical board, circuit board, which will provide the supply inside the cabinet. This is the power supply of a this is also a kind of an auto pipette. This is actually a SteriPet. So you can see in the front of the camera, this is the SteriPet. This one is for taking up, and this one is for taking down. This you can use for putting the culture medias, and you can also different types of solutions from here. This is a Tarson SteriPet. So this is the main things you require, the minimum things you require. These are the tubes, some different types of sample collecting. These tubes you can use for sample collecting. You can use this for keeping mycotoxins. If you can use for culture media, anything you can use. So these are the main things, and above all you require some flasks and Petri dishes that we will be seeing in the next part of the video. Some flasks and culture flasks and tubes. You can see in here, you can see these are the culture tubes. These are the small culture tubes. You can see these are all 10 ml culture tubes. These are very easily available. So these are the culture tubes, and this is also a special type of culture tube. This is 30 ml borosilicate glass. This is made up of 30 ml borosilicate glass. As you can see, this is a 30 ml tube. This is also used for culture media. These are the glass tubes of 20 ml. These are the glass tubes of 20 ml. This also you can use as a culture media, and you can use a 50 ml flasks also, that you have seen in my videos, previous videos. We will see now in the next part of the video. We will see now regarding the culture media and the apparatus. Now in next part of the video we will see the medium, different kinds of apparatus, and the uh, things, small things, the apparatus, petri dishes and all that you will be able to see. So let us go for the next part of the video. So you can see in the petri dishes, two petri dishes are here, and a 50 ml culture flask. This is a small 50 ml culture flask. This you can see. So actually, these are required for the culture media. Petri dishes are mainly used for you can keep it for culture media also, or you can use it as for viewing the under TCM 400, inverted TCM 400 microscope, for direct investigation of the samples under microscope. Just from taking the some flasks, some culture media around the culture media, and taking them in mass, and you can see just in the drop in the petri dish, and you can just see before the under TCM 400 microscope. For direct investigation, these petri dishes are used. So regarding this petri dish, you can see two types of petri dishes are there. One is this one, and the other is this one. What is the difference between these two? If you see the edges very closely, if you see the edges, just see the edges, see the edge of this, and see the edge of this. So this is much thicker. This petri dish is much thicker. So what is the problem in this? The problem is, if you use a very thin petri dish or a very sleek petri dish like this with a low edge, then your vision will be correct. You will have a very correct vision under tcm microscope. It will be very visible to see, because tcm 400 microscope, actually what it does, it actually sees from the background, from the bottom, and the light falls from the up. Your sample is here, the TCM, you will place this on the TCM 400 microscope. This will be placed over the TCM 400 microscope. Suppose, let us assume that this is the stage of the petri dish. This is the stage of the assume this box is the stage of the TCM 400 microscope, and you have to place the petri dish over that. So light is, the objective is inside this box, and the light is falling from the top. So if what happens, if this glass is very thick, then what will happen, the microscope will be very difficult, will face a difficulty regarding this focusing. So you won't be the image will be blurred. So therefore we have to use this very, the as much as thin the edge of the petri dish would be, that would be better. So thinner the edge, the focusing will be easier, and the vision will be clear. The resolution of the picture will be very clear. Resolution of the image will be very clear. So these petri dishes are very old petri dishes. Nowadays these petri dishes are used. These are actually, you can see, if you see, these are actually Borosil petri dishes. This is the original Borosil S-line petri dish. So it's a slim line petri dish. So this type of petri dishes are very good for investigation under inverted tissue culture microscope. And this is the culture flask, just we can use so for culture media. So this we need, these are the basic instruments we need to, apparatus we need to do the microbiological investment. This is the start point. So this, you have to have this type of instruments or this type of apparatus first of all, to make the start the culture media, or you can start the fungal culture or the bacterial culture or whatever it is, any microbial culture. You need to have this must in your lab. So this is your actual start point of the microbiological lab. So total making this will be around how much, around 5 to 10,000 rupees you can make. Regarding the steady pet and those autopipettes, a little bit costly. Otherwise, if you leave those prices, otherwise you can have at least 10 15,000 rupees you can make. You can make within that, you can make this total microbiological cabinet. So in the next video we will going to discuss about the culture media and some other techniques, how to make the culture media and what type of culture media we should use. that we are going to we're going to study in the next video. So off from now. Thank you very much.